Hello and welcome to some lovely research methods. We're doing the second part, aren't we? Part 2. Very good, Sarah. Good, Sarah. Have a gold star.
Yay.
We love gold stars. Today we're going to fit in the middle part. There will be a third video, trust me. We're going to kick off with qualitative and quantitative data because peeps get them mixed up and shiz, doesn't they? They does.
Yeah.
Quantitative data is in the form of numbers, e.g. 56% of people like onions. It's easy to analyse, it's objective and less open to bias, but less depth is obtained. However, it's just saying it's in a numerical form won't get you the marks. You've got to say it's in a comparable form. You can compare it to other things. That's what you've got to say. Something along those lines. Qualitative data is data that in a non-numerical form. For example, a transcript of an interview with an alcoholic explaining why he drinks. Weird. Don't drink, kids. It allows a detailed response and can be used to express feelings. However, it's open to bias and difficult to analyse.
Yeah.
Right, on to aims and hypotheses. A hypothesis is a testable statement. A null hypothesis predicts that there is no effect on the IV or the DV, or no difference between condition 1 and condition 2 in whatever. An example, listening to classical music will have no effect on the number of clues correctly solved in a crossword puzzle in 5 minutes.
The alternative or experimental hypothesis predicts that the IV affects the DV.
A directional hypothesis predicts the direction of the effect, obviously. Like listening to classical music will increase the number of clues correctly solved in a crossword puzzle in 5 minutes.
A non-directional hypothesis predicts a difference but not in a certain direction. E.g., listening to classical music will affect the number of clues correctly solved in a crossword puzzle in 5 minutes.
Also known as tailed and two-tailed.
Two-tailed.
Or one-tailed or
and
two-tailed. In a particular study, you will have a null hypothesis and then just one alternative. When deciding whether it should be directional or non-directional, look at previous research. If previous research shows the direction of the effect, use a directional hypothesis. It's that simple. Note for correlations, the word effect cannot be used, and use the word relationship or correlation like there were. For a null, there is no relationship between intelligence and exam success. An alternative, there is a relationship between intelligence and exam success, that's non-directional, and there is a positive correlation between intelligence and exam success. Or, that's it. Note, in the exam, hypotheses should be fully operationalised in detail. This means that they should be as specific as possible. Use the exam question to help you. For example, the effects of classical music on concentration levels is being studied. The concentration is being measured by the number of crossword clues solved in 5 minutes. Does that make sense?
Mm-hmm.
Sort of. Just make sure it's got numbers in it, really, or some sort of measurableness.
Yeah, lots of detail.
Lots of detail. Fill up the space provided. Build up the null hypothesis. Music has no effect on concentration. Then fully operationalise it. The null would be listening to classical music will have no effect on the number of clues correctly solved on a crossword puzzle in 5 minutes. See? Sorted. Right, on to research designs now. 3 types. Repeated measures design, where you have the same participants in both conditions. Independent measures design, where...
different participants are used in each condition.
And match pairs, where...
different but similar participants are used in each condition, and effort is made to match the participants on important characteristics that may affect performance.
Strengths of repeated measures is that participant variables or individual differences are kept constant. So intelligence, past experience, and more powerful statistics can be used, and fewer participants are required, so it could be cheaper, is a nice little thing. Weaknesses is order effects may occur. For example, they might, if they practiced it or if they're bored the second time round. Counterbalancing, though, can be used. So half do condition A first and then half do condition B first. So like that, with the same people. Increased demand characteristics, because participants do both conditions, they may guess the aim or act and therefore act differently, and different tests may need to be, just may be needed.
Strengths of independent measures, you have order effects do not occur as each participant is only in one condition, and demand characteristics is less of a problem as they only do one condition again, and the same test can be used. However, weaknesses are that participants' variables are different, therefore possible, e.g. one group are more intelligent than the other, therefore you have confounding variables. And another weakness is more participants are required.
Strengths of matched pairs. Participant variables are kept constant, no order effects, less demand characteristics, and the same test can be used. Weaknesses is that participant variables can never be matched in all respects. Matching participants is time consuming and difficult, and more participants are required. So again, it could be more expensive. Pilot studies now. I love pilot studies. Pilot study is just a small-scale trial run of the actual study that is done so that any problems with the procedure can be adjusted before time and money are spent on the full-scale study. You will need to know that exact definition. Just remember to put procedure in there, really. Standardised instructions, that's a set of instructions read out to the participants to ensure that all participants give the same information. It's a good idea to put that in a written form or have it in a video so that everyone gets the same instructions.
Yes.
Observer checklist. When a different observer carrying out an observation must make sure that they have a clear description of what behaviours they are looking for and how to score them. For example, aggression checklists may include the number of times hit another person, the number of times spat at another person, etc. It may use examples to make sure they're all working to the same standard, and it therefore ensures higher inter-observer reliability.
Operationalization of the DV. Defining the DV in terms of a measurable task. For example, boys are more aggressive than girls. Aggression measured as a number of times hit another child in 15 minutes of play.
Pilot studies can find out any possible problems with the procedures before they invest huge amounts of time and money, and any problems can be adjusted, as I've already said. In an experiment, you can identify the problems with the standardised instructions and can check the suitability of the measures of a DV. In observations, you can check whether the behaviour checklist is appropriate and then check the training of the observers to ensure inter-observer reliability. And in a questionnaire and surveys, you can check the interpretation of the questions and make sure they're not ambiguous. Check the standardised instructions are clear and make sure they're not too complex questions, too simple questions or confusing questions. And that's why we put that all under pilot studies, which kind of is. The relationship between the researcher and the participants now. This is always forgotten. I don't really know why. Kicking off. Demand characteristics, cues or clues in an investigation that convey the researcher's hypothesis to the participant. In doing so, they influence the participant's behaviour. When entering a lab, participants try to make sense of the situation, and often this involves trying to guess the researcher's hypothesis. Investigator effects.
And the various ways in which researchers influence the behaviour of participants.
Expectancy effects. Researchers are, this is under investigator effects. Researchers are human beings with their own hopes and expectations that their experiment will work. As a result, they may influence the behaviour of the participants. Usually, they are not aware of doing this, but they may give away using body language or their tone of voice, etc., and which the participants may pick up on.
Yes, and then you also have individual differences, where no two researchers are the same. They differ in age, sex, ethnicity, social class, etc. To some extent, their personal and social characteristics will have some effect on the behaviour of the participants. For example, female researchers are more friendly to male participants, and black participants may give more detailed answers to a black researcher.
That's all investigator effects. Participant effects is separated to 3 bits. Firstly, evaluation of apprehension. This refers to the concern felt by participants that they are being judged or evaluated by the researcher. Many participants want to be seen in the best possible light and therefore try to hide certain aspects of the behaviour that they think it may be interpreted in a negative way.
And participant reactivity. This refers to a situation in which an independent variable has an effect on the behaviour simply because the participants know that they are being watched.
And the third participant effect is individual differences again. Participants also vary in terms of their personal and social characteristics, and these can affect their behaviour. For example, some participants like to please the researcher, and others like to do the opposite, the screw you effect. Controlling demand characteristics, investigator effects, and participant effects can be done in 4 ways. Firstly, blind procedures. In some studies, participants know the general aim of the research. However, demand characteristics can be reduced by concealing from the participants which condition they are in. This is known as a single blind procedure. However, it does not stop the participants guessing and altering their behaviour. In a drug studies, for example, a placebo is used where the group not given the drug are given a sugar pill to overcome expectancy effects. With a double-blind procedure, research assistants are used to direct the experiment, and neither they nor the participants know which condition the participants are in. This technique is often used in drug studies.
Yes, and now deception. One of the most common techniques used to reduce demand characteristics is to deceive participants. For example, in Milgram's obedience studies, participants thought they were doing a study on memory, and most researchers think that this blatant deception is unethical, but in some circumstances it's deemed necessary to withhold the aim from the participants.
Automation. One of the most effective ways to reduce investigator effects, actually, is to remove the researcher from the scene. For example, instructions can be tape recorded or presented on a computer screen using a camera for observations. Remember to install several weeks before to start recording behaviour so participants get used to it and act naturally. And finally, alternative research method.
Most of the effects mentioned apply mostly to lab experiments where participants are aware that they are being watched and communicate directly with the researcher. Often field and natural experiments avoid some of the problems.
Finally, last little bit in this video, selection of participants. Don't use more than 100 participants, but generalise findings to the whole population. The participants are called a sample, if that is the case. So, if a sample is taken from a large population called the target population, which consists of all members of the group which the sample has been taken, so which you're studying. In order to be able to generalise, the sample must be representative, i.e. it must be typical of the whole population. If it is not representative, then the sample is biased. The sample size is important because if it's too small, it will not be representative, and if it's too big, it will be expensive and time-consuming. Usually about 30 experiments. That's how many you should have. Where a small effect is being investigated, for example, correlations, then large samples must be used. Methods of sampling. First one.
Random sampling only occurs when every member of the target population has an equal chance of being selected.
Stratified sampling
involves dividing the target population into important subcategories and then selecting members of these categories in the correct proportions. For example, if the target population is a school, we might select members of each year group in correct proportions.
Well, you won't ever be asked about that, but you can mention it. Mm-hmm. Opportunity sampling.
Select participants that are available at the time. For example, universities usually use psychology students.
And self-selecting or volunteer sampling.
Individuals or volunteers who consciously or unconsciously determined their involvement in the study. Unconsciously, for example, observation in the street.
Strengths of a random sample is that it provides the best chance of an unbiased representative sample of the target population. Weaknesses, though, is compiling a list of everyone in the target population is impractical, time-consuming, expensive, and therefore it is very rare. The strengths of a stratified sampling, it's a deliberate effort is made to identify the characteristics of a sample for it to be representative of the target population. But the weaknesses, it can be difficult to identify important characteristics and again can be very time consuming. Strengths of an opportunity sample, it's quick, convenient and economical compared to the other methods. But a weakness is it can be unrepresentative and often biased on the part of the researcher to pick helpful participants. Strengths of a volunteer sampling is it's convenient and if volunteering is made in terms of informed consent it is ethical. But weakness is its unrepresentative samples. Volunteers are often different, for example more outgoing, helpful, higher intelligence than typical members of the population. That's it for this video, again in just under 14 minutes.
Woohoo!
Yay, we have to fill 15 seconds now. Just filling it with silence, 10 seconds. Bye. Bye. Say bye.
Bye.